Good morning, everybody. It's an honor to be part of the formal symposium. I wore my coat, I wore my pants, and I did it to not embarrass my wife, who didn't show up. All right. I've already probably used too much of my time. So our lab is interested in fruit development, and particularly in fruit ripening, that kind of end stage of maturation, where the fruit does something really kind of amazing. It does what the plant is not trying to do its whole life, which normally it's trying to protect itself from infection, from having things eat it, and it undergoes this transition where it suddenly becomes succulent, attractive, aromatic, nutritious, and basically says, come and eat me. And who does that? Frugivores, so animals that eat fruits, and yes, it's a funny slide, and in 6 minutes, it's the only thing you'll remember. And disperse the seeds, right? So this is particularly for fleshy fruits, and we're interested in understanding, what controls that process of that very dramatic developmental change. So I was going to point out to the interns who, of course, aren't here since their program's over, always good to put a acknowledgement slide early before you run out of time and forget to acknowledge people. This is meant to acknowledge everybody who's been in the lab for 32 years. So those of you who were here before Dan are not on the slide, but you're there in spirit. And also to point out that often in labs, you've got someone with a really big head who is supported by all the people who do the work behind them. So what's ripening? Well, it's not just color change, which is often what we see and think of most, but it's all these different biochemical processes and physiological processes that are coordinated somehow to bring about this dramatic change in the fruit. And while different fruit do different things, often they convert their starch to sugars, they change in texture, they get soft, cell walls break down, pigments accumulate, sometimes the chlorophyll degrades, sometimes it doesn't. Aroma volatiles are produced, protective compounds like glycoalkaloids that normally keep things from eating them are reduced. They generally become susceptible to opportunistic pathogens, largely due to that breakdown of the cell walls. And in many, but not all fruits, the hormone ethylene is critically involved. And if 20 years ago you look at the literature, the control of ripening is ethylene. And we didn't know much more about it before that. So that's what we've really been interested in. What's upstream of ethylene? What controls ripening in fruits that don't seem to use ethylene for their ripening? And why is it important? Because unlike many of our staples that are often seeds or tubers that are evolved to overwinter and persist and therefore are relatively easy to store and preserve at low cost, fruits do just the opposite. They break down, they rot fast, and as a result, you lose a lot of your potential yield and a lot of your potential food and nutritional security, particularly in parts of the world where you don't have access to systems to have controlled environments and maintain things. So it's a big problem. And we spend a lot of time and energy trying to control this process, and understanding it is key to control. So what do we do? Over the years, many different things. We've looked at naturally occurring mutants whose phenotypes tell us that there are underlying genes that control process. And we've isolated many of those genes. We take a number of different mapping approaches to identify additional loci. We do way too much transcriptional profiling to identify additional genes whose expression is associated with ripening. And we've been looking at the epigenome and how it essentially puts a block on ripening until the seeds are mature. And we also do a lot of functional analyses, a lot of RNAi, a lot of CRISPR, and after Dan's beautiful talk yesterday, hoping this gets much easier in tomato soon. More recently, we've been taking advantage of the incredible and way underutilized genetic diversity of tomato. And really to do two things. One, build a tool that'll allow us to access it more in terms of connecting structural variants and changes in gene sequence with functions, and then to deploy that tool for our interest in fruit ripening. And that's really what Anna Hermans, the PhD student who will talk later, will talk to you about. She'll focus on this part of our work. And I'll just end with tomato is lots of fun. You'll never see a picture like this with Arabidopsis. Thank you.